All right. So today I'm going to talk about kidney microvascular endothelium. And when I say microvascular, I'm just talking about the capillaries in the kidney. And I'm going to talk specifically about their role in peripheral T cell tolerance. And this is a story of my research, an abridged version. And I'm really going to focus on the significance and interpretation of decades let me just get this out so I can see, decades of data. So the outline I have here, but the bottom line that, sorry, I just want to get this out so I can see. The bottom line is that I propose that the kidney microvascular endothelial, via their HLA class II expression, creates an immune system that contributes to peripheral T cell tolerance via something called programmed death 1 ligands and other inhibitory signals. And that's a whole lot of immunology language that probably nobody really cares about, but I want to tell you that this really is an interesting system. But we're going to start off with some definitions and concepts. And the first one is that of tolerance. And tolerance just means that you have immunological unresponsiveness. So we don't want to respond to autoantibodies. We would love it if kidney transplants, we could, make them so that we're tolerant to them. But you do want to respond to things like cancers and infection. And in terms of T cell tolerance, there is a central mechanism whereby immature cells that recognize antigens of high avidity in the thymus die. And in the thymus, you actually get a whole lot of peripheral proteins expressed. So nephrin is expressed in the thymus. So what happens is that any T cell that would see this nephrin would be essentially deleted. They call it negative selection. And when you're young, you have a robust thymus. Here's these immature T cells that get deleted. And when you're old, ancient like me, you're lucky if you have this much left and there's no more immature T cells. So does this mean that at my ancient age that I no longer have a mechanism for getting rid of reactive cells? Well, not really. There is a second form of tolerance that comes in the periphery, and it's more of an anergy. And what I mean by that is that the mature T cells exposed to antigen don't respond. They either don't have the right co-stimulation, there's an inhibitor, there's regulatory T cells, they become inactive, but they don't die. Unlike with central tolerance, the cells are actually wiped out. So if you can understand how this anergy occurs, the idea is maybe we can harvest it to turn off T cells in autoimmune conditions and transplants and increase it to kill cancer cells. And manipulating anergy is a clinical reality. This is what we do when we use these things called checkpoint inhibitors, most of which are antibodies to part of this programmed death pathway. They block essentially anergic signals so the T cells can go and kill the cancer. And this was approved in 2016, so we have now been using it for 7 years or more. The next concept I want to go over is how T cells recognize an antigen. So you have an antigen presenting cell. It could be any nucleated cell. It has an HLA molecule on it. There's a peptide that fits into that HLA molecule. And then the T cell with its receptor sees the peptide and the HLA molecule, and it can go on to proliferate, release cytokines. You can look at various activation markers when this combination occurs. If you don't have a peptide, there's no response. If you got the wrong HLA molecule, there's no response. And if you have an empty T cell receptor, there's no response. Now, in the antigen presenting cell, they can express HLA class I proteins designated A, B, and C. And those are the ones that activate CD8 T cells. And they have HLA class II proteins, which are on a limited number of cells. They used to say just B cells, macrophages, but I'm going to tell you that they're on endothelial cells, and they activate the CD4 cells. And this is going to be the focus of the research that I'm going to share with you today. So those are my definitions and concepts. And now I want to give you an abridged story of my research. And the story begins with Bill Kauser, who was division head at the time. And Bill encouraged science from other disciplines. He recognized that dialysis was here and that nephrology was probably going to die if we didn't come up with some other more basic science and some other things within our discipline. And so I looked around for labs to work in when I was a fellow and came upon that of Don Pius, who was a pediatrician. And he had some neat things going on where he used genetic mutations, some he made, some that were that nature made, to look at the regulation of these class 2 genes and how that antigen peptide actually got processed to fit in the molecules. And all of his work was done with humans. And there I met this microscopist, Susan Anderson. And she was been in the university a long time and probably one of the best microscopists we had. So we had these reagents for class 2, and Susan said, go get me some kidney and let's see if we can have and find any of these class 2 proteins in the kidney. Now you can use an antibody to class 2 proteins that's specific for a certain allele, or you have some antibodies that will look at all of, say, the DRs or all of the DQs and DPs. And when we did this, this is what she found. Very bright staining in the interstitium and the glomerulus, but not in the large vessels of the kidney. And when she put a co-staining antibody there for endothelial cells, the pattern was the same. So that if you merge the image, indeed, what you found is that there was very high expression of this HLA-DR on endothelium. And so these kidney microvascular endothelial cells, but not those of the larger vessels, express HLA-DR. And this was scary to me because it shouldn't really happen. And I'll tell you why. And that has to do with how this peptide gets there. The peptide for class 2 comes from the uptake of extracellular proteins and peptides. So if this endothelial cells are expressing this, it means that any protein circulating by those blood vessels can potentially become an antigenic peptide. And just to emphasize again, here's your HLA class 2 pathway for antigen presentation. And what happens is you get a protein that's endocytosed, it gets broken down into peptide, and then it gets bound on your HLA molecule to present to a CD4 T cell. HLA class 1 proteins come from actually intracellular proteins. So a cell gets infected with a virus, the virus gets chopped up into a bunch of little peptides, and then it comes out here and is presented to a CD8 T cell. But the microvascular endothelial cells express these class 2 molecules, and they're therefore, because they're in circulation, have the ability potentially to present any protein or peptide that's circulating by them. Now, the endothelium expresses these molecules, these HLA-DR, and they can encounter circulating T cells, but there's no inflammation in normal kidneys. You don't see a kidney inflamed, even though these proteins are there. And maybe it's because all those autoreactive T cells were deleted in the thymus, so we don't have anything left, that the thymus is that good about getting rid of them. This does not occur in rodents, and most of immunology is all based on mice, so you don't see this in the textbooks. And of course, when I made this initial observation, I thought it must be wrong because it's not in the textbooks and nobody's talking about it. But then immunologists aren't really dealing with humans for the most part at this point in time. So I wanted to study a little bit more about how this HLA business gets out on these endothelial cells, how it's regulated. And I'm really interested in whether or not it could present peptide to activate T cells. Now, in order to do this, I had to develop a technique where I could go from tissue to cells so I could actually study the cells. And this took several years, but I chose to use flow cytometry. And what you're looking at here is I have a digested tissue, and each one of these dots represents a cell. And here, these cells expressing high DR and an endothelial marker are the microvascular endothelial cells. And if I want, I can say which ones of those are from the glomerulus and peritubular area based on different markers. And I can also sort out over here the larger endothelial cells that don't express DR. So I now had this technique to identify these cells. And the first thing I wanted to do was to ask about the regulation. How does DR get out there? Normally, it's thought that class 2 is only on macrophages and only on B cells, and that it can be induced with gamma interferon. And a key component is that you have to have this transcriptional factor called C2TA. It's a non-DNA binding protein that gathers up a bunch of other proteins on the gene and can go ahead and then make class 2. And I'm going to skip through this because it first last of time. But the question I had was, do I have C2TA in these microvascular endothelial cells? And I will skip through these because we're going to run short of time otherwise. And the bottom line here is that I do have the C2 promoter, I do have the C2TA, and it's and I do have it, but it requires a very low physiological concentration of gamma interferon. So these endothelial cells express DR, they have the regulation C2TA, but it does require a physiological level of interferon to turn it on. Not what's in inflammation, but what's normally in circulation. Now, the other question that is more significant to today's discussion is, can these endothelial cells actually present a peptide to activate T cells? And thanks to Jane Buckner at the Benaroya Research at Virginia Mason, she gave me some T cells that recognize influenza peptide when it's presented by this particular DR allele. So this is the antigen presenting cell, and we're going to look at some proliferation data, which is marked by uptake of tritiated thymidine. A cell proliferates, it takes up tritiated thymidine. So here's my endothelial cells of the right DR type. I don't add anything to them, not much. I add peptide, not much. T cells, not much. But if you put the peptide, the T cells, and the endothelial cells together, you get proliferation. If I choose an endothelial cell that I have isolated from a patient that isn't of this DR type, I don't get proliferation. And just as a positive control with the B cell that has lots of class 2 on it of the right HLA type, you get a lot of proliferation there as well. So the endothelial cells can cause proliferation of specific T cells with peptide, and that's with the appropriate DR restriction. I did some other studies, blocking studies, to see how this might be controlled. So again, here's my endothelial cells. Adding T cells, you don't get much. You add peptide and T cells, and again, you get some proliferation. If I put an antibody in that's going to block all the DRs, it goes away. If I put an antibody in that blocks something called CD58, it all goes away. And holy smokes, when I add an antibody that blocks this programmed death, I get a lot more activation, a lot more proliferation. And at the time I did these studies, nobody even really knew what these antibodies were. I actually got it from somebody in a lab, and they said, oh, just try this. And it was really quite amazing. And the same thing if you want to look at gamma interferon that T cells make when they're activated. Here's my T cells with the peptide. And if I block here with this PD-1, you get a lot more cytokine release. So again, these endothelial cells can activate T cells. It requires the DR, I've shown that. It requires this co-stimulatory marker. And if you block it, you get enhanced activation. Now, what is this programmed death thing, this PD-1? And, you know, all the immunology, they have all these CD things. So, you know, PD-1 has got a designation of cluster differentiation of 279. So here is my T cell. Here's the T cell receptor. MHC is just the HLA molecule. And what happens in order to get this signal, this is the recognition, the T cell receptor has to get activated by kinases that add on a lot of phosphates. This PD-1 here on the T cell with the ligands, if they get activated, they turn on a bunch of phosphatases to remove the phosphate. So that the PD-1 with its ligands recruit phosphatases to inactivate this T cell receptor, which requires to have phosphates there. So the summary at this point is that the endothelial cells, or microvascular endothelials in the kidney, express HLA class 2 with physiological concentrations of gamma interferon. They can activate T cells in this DR restricted peptide dependent manner, but this T cell activation can be limited by the engagement of this PD-1 pathway. Now, when T cells see an HLA molecule with a peptide, that's a very high recognition signal, but they don't respond. They need some kind of secondary signal. And so I tried to look at what those might be within these endothelial cells, again, using my flow cytometry technique I had developed. So what I did is I took kidneys, native kidneys, normal kidneys, got them digested up, selected these cells, and asked, what are the secondary signals on these endothelial cells? And a complicated slide that summarizes a lot of data So here's my endothelial cell. This is actually a T cell within the kidney. And everything in green are the activating secondary signals. And in red, the inhibitory secondary signals. So signal one, the endothelial cell has to recognize the T cell, and then you have to have one of these signals to turn it on. And where there's open areas here, it means I didn't find those molecules. So this was, say, B1 or B7, 1 and 2 are activating for certain kinds of T cells, but they're not present on the endothelial cell. So that's why those are open. You'll notice here that you do have this CD58 and CD2 binds the CD2. This is the arrow means this is a pair that when they bind, they're activated. And again, when I blocked this, my activation went away. And then look at all these inhibitory signals on the endothelial cells. You have two ligands for these programmed death one, and you have two other inhibitory signals that we don't even know what they pair with, but in vitro data says that they will inhibit T cells. I also point out on the kidney T cells, here's this PD-1. Now, in order to make this work, I've got to have this receptor here. And looking over here, this shows a positivity. Not all the T cells in the kidney express this, only some. And those that do then can be turned off. And that'll become pertinent later. So 3 inhibitory secondary signals are expressed on the kidney microvascular endothelium. We have this PD-1 system, and we have 2 others, something designated B7H3 and B7H4, that we really don't know what the receptors. All of these are now being investigated as cancer therapeutics, because what you want to do is get rid of the inhibition so the T cells will go kill the cancer. Similar observations have been made on microvascular endothelium and other organs, but not studied to this extent. So there's similar findings in the heart, where the capillaries have high HLA-DR. They can signal T cells via this CD58, and they have all the machinery for putting a peptide out there. And there's some similar things in the gut. So what this raises as an issue is, does the microvascular endothelium, with its HLA class II expression, represent some human vascular immune system that can result in anergy to turn T cells off? I also took a look at some things in development. We have a great lab here, the Birth Defects Research Lab, that I was able to get some fetal kidneys and look at essentially their microvascular endothelial cells, which I've shown here. And what it turns out is that the HLA-DR, this class II, is expressed on endothelial cells in development as early as 60 days. And does it have these ligands, these inhibitory ligands? Yes, it does. These are the inhibitory signals which are positive, so that most of the endothelial cells in early development with HLA-DR also have the ability to turn it off via this programmed death one system. So I interpret this as that the expression of this HLA-DR with the ligands to the PD-1 on the microvascular endothelium early in development really does, I think, support the idea that there's an endothelial-based immune system that's not necessarily induced by inflammation, but it happens early in development and probably has a significant role in anergy. All right, so I was doing all of these studies in T cell activity with isolated cells. But the key thing is, let's have a human experiment. And that comes when we look at what happens when people are treated with this PD-1 blockade reagents. So you block this PD-1 pathway in humans, and I do think the oncologists who have been using a lot of this, and we know that you can get some toxicity. And the toxicity that you get in the kidney is an acute kidney injury. Here's 2 examples shown with nivolumab, which block the PD-1 pathway. And what you notice is that you have this acute interstitial nephritis, mononuclear. You don't see eosinophils like you would in an allergic interstitial nephritis. The gloms are okay. There's no immune complex. And you can probably get similar findings in other organs. Again, this microvascular endothelial system that I'm studying in the kidney exists in other organs, I predict. And I propose that what's happening here is that this inflammation comes because the microvascular endothelium HLA class II is seeing T cells that now no longer have the blockade of PD-1, just like what I did with culture. So what are the predominant cells infiltrating the kidney with this acute kidney injury that comes with these checkpoint inhibitors? Well, It's T cells, and that's what you would expect. So here you're standing for T cells by CD3, B cells, a few there. CD8 is a marker for monocytes, granulocytes. It actually can label a lot of cells, but the T cells really predominate. So again, we have toxicity in the human when we block these PD-1 signals, but it doesn't happen in all patients, because what you have to have is that first recognition signal. You have to have a T cell that is actually seeing an HLA molecule, but if it does, and you don't have any way of turning it off, it's not such a good thing. So what about some implications for autoimmune disease and transplant? We checkpoint in modulation. In cancers, what we want to do is we want to block these inhibitory signals. You want the T cell to go get the cancer. But to treat or prevent rejection or autoimmune disorders, we actually want to enhance these inhibitory signals. We would like the T cells turned off. And so you can consider that in a transplant setting, would a transient enhancement of inhibitory secondary signals on the allograft endothelium, could that make the T cells anergic? Right now, what we do is we just wipe out the T cells, but maybe what we should do is make them so they don't see the HLA molecules on the endothelium. And so again, the idea is if you had a kidney allograft, what you're trying to do is to turn off some of these signals that are activators, and what you want to do is turn on these inhibitors. And they're actually beginning ways of doing this. So there is now some peptides that can block this pathway here. And what you're going to find is that there's a lot more development of molecules that are going to try to enhance some of the inhibitory signals. There's this peptide I mentioned. There's other things that are being used that will stimulate the programmed death system for rheumatoid arthritis. There's people that are mapping out on PD-1. If you have an antibody to one part of the molecule, it's an inhibitor. On the other hand, it's an activator. And there's some really cool bifunctional things where you can sort of tie a T cell receptor to the programmed death agonist to inhibit T cells in a more specific way. And if you don't think this is real, if you look at the number of companies that are now looking at secondary signal checkpoint agonists that are coming in the pipeline. So instead of just for the cancer drugs, what we're going to do is to try to turn on the inhibitory signals for things like rheumatoid arthritis and some other autoimmune conditions. Stimulating energy is a clinical reality that's just begun. We use these checkpoint inhibitors right now primarily to program death pathway things, to block these anergic signals to cancer cells. And I think what we're going to do is find that there's going to be some agonists that would be useful for other mechanisms of disease where you actually want more inhibition. So now I need to give you my sort of interpretation and biological speculation of this. And one of the things that's really weird is why have secondary signals on endothelial cells that both stimulate and inhibit the T cells? Why don't we just like turn any T cell off that we recognize and not stimulate them at all? So this kind of comes from some other experiments I did with T cell assays. And I wanted to try to bypass the need for having a specific peptide and HLA molecule. I mean, it's hard to get the donors of the right HLA type, and you have to get specialized T cells. So let me just change my assay around. And what I did, instead of trying to use this signal 1, which I had done with the influenza peptide, let's just change this and activate the T cell by using an anti-CD3 molecule, which will trigger the T cells. And let me just use some peripheral blood T cells. You know, why should I get anything special? And let me go ahead and stimulate these cells and then try to inhibit some of these molecules or the activating molecules and see what I get. And here's an example here. And what I'm looking at in this readout is not cytokines or proliferation, but just the appearance of an activation molecule, something called CD69. And here's my T cells. This is a renal microvascular endothelial cell that got changed to a kidney microvascular endothelial cell in later years, and the CD3 stimulus. There's my blind activity. I can put in antibodies to the CD58 and its receptor, CD2, it goes down. And look what happens when I block this PD-1 pathway or one of these other inhibitory molecules called B7H4. I get a lot more activation. But I get that activation only in memory cells. cells that have been previously activated, and this 45RO is just a signal of memory cells. So it turns out that PD-1 is present only on activated T cells. Most of the T cells in peripheral blood don't express it. So you need some sort of T cell activation to induce this PD-1 expression so that then the endothelial cells can make them anergic or unresponsive. So you go back here to this, and here's my endothelial cell. They have always turned on high expressions of these ligands for programmed death. If I looked at the kidney T cells, only some of them express this PD-1. And if you look at peripheral blood, there's even less. So the concept is, if these guys are going to turn off the T cells, they have to have T cells that express the PD-1 or these other unknown ligands, which we have yet to figure out. So the biological sense of having secondary signals on the endothelial cells that can both stimulate and inhibit T cells, in my mind, needs to be there to provide what I'll call an activation trickle to the T cells, so that those T cells that recognize a class 2 peptide on the endothelial cell can also then express these inhibitory signals. And then with a small amount of activation, you can actually make them anergic, because you can then turn it off by the PD-1 pathway, and you can save yourself having a massive inflammatory response. Some other comments about biological significance of this system, which is present early in development, is why do we have this, And in a way, maybe it's to protect the parenchymal cells behind the endothelium, that the endothelium can take up some debris that might be recognized by T cells, and it's evolved this mechanism to make the T cells anergic to it. And the other issue comes up is we always think the thymus does all the work getting rid of our autoreactive T cells, but perhaps there is more autoreactive T cells that really escape this thymic deletion than we appreciate. And this microvascular endothelium system could be the one that tames these circulating autoreactive T cells that fail to get deleted. So the conclusion is that the kidney microvascular endothelial cell, via this class 2 HLA-DR, creates an immune system that I think contributes to this peripheral T cell tolerance via the PD-1 ligands and other inhibitory signals. And with that, I want to acknowledge the many people who have helped along the way. And I thank you for listening to my journey, and I'll take any questions or any thoughts that you have.
Thank you, Kim. Thanks for presenting this, what I know is only a small part of a huge body of work. Questions for Kim?
I recognize this is not an immunology audience, but immunology is what where a lot of our disease comes from, if not all.
Well, with that invitation to that you're speaking to a clinical audience, can you so sort of hinting at some of these pathways maybe being clinically relevant in the future for transplant immunosuppression, can you kind of put into context where potential medications really?
Well, let's say you get a kidney transplant. Right now, what we do is we just block out all the T cells, get rid of them, eliminate them. But maybe what we should do is actually put in some of these inhibitory signals so that the T cells will have a little activation, and then you just shut them off so that they no longer cause a problem. Or maybe if you have rejection when those cells come through, you again put in some of these inhibitors, you try to increase it so you can shut them off. And so it's a little different than wiping out the immune system totally, because you could probably use them transiently, because we think that once the T cell is anergic, even though it's still there, it probably is going to be anergic thereafter.
And other components of the current sort of regimen for transplant that you'd anticipate this would kind of this would be upstream or downstream, would be more specific versus
I think one of the things that I would think about first is whether or not they would have any role in rejection. But you got to admit, we're pretty good with our immunosuppression now. Nobody's going to want to futz around it. I do think that it gives us another thing to look at. And I think you're also going to find for some, perhaps an autoimmune disease, wherever there's a T cell component, as we learn more about that, it gives us a whole nother way of manipulating the system. quite frankly, cancer gets a lot more money than nephrology. So that's why they focused on using these to get rid of the cancers by immunological means. And, they're very, very effective. I mean, a lot of metastatic disease, is really arrested by going ahead and getting rid of the inhibition so that, you make your T cells just take care of the cancer. It's just that we, are coming into side effects because I think the endothelial cells need this so that they don't become reactive.
Very characteristically economical of you to borrow from cancer and pull it in. A question from Ashley, if you can see the chat, but are there HLA-DR genotypes that increase the risk of PD-1 inhibitor-associated tubulointerstitial nephritis? Does that tell us anything about potential antigens, peptide sequences?
No, not that I know of. I mean, any of the DRs will work for this. And, I haven't typed all of the donors that I have, but, I've got a variety of different donor types. It also, I think, it is similar. And in the endothelium, it's not that just the PD-1 ligands are there on a few cells. They're there on all cells. It's the PD-1 receptor on T cells that is not present on all the T cells. So you have to have that in order to make the system work. And again, there are other inhibitory molecules, that B7H3 and H4. Nobody knows what their receptors are. And so I think, if you did that, that also gives you another way of trying to manipulate the system.
Hi, Kim, this is Yudoya. Hi, really great talk. I always enjoy listening to you. So I have a question. You were talking about DR expression on microendothelial cells. Have you also looked into DQs and DPs also?
Yes, it's there. It's there at a lower amount so that, your signals aren't as high, but they all match. You have the class 2 transactivating C2TA and DP and DQ are there. But I just have done DR because it's the most abundant.
And do they respond to inflammation, meaning increase expression under inflammation, or they stay stable? The expression.
Do they increase with inflammation?
Yeah, so do they respond also to increase the level of expression? Like we always say that under inflammation,
right? I don't think so.
So they are
I don't think so
expression.
Because I've looked at lots of biopsies from Nikolai. And even in the context of rejection, you're going to get the same level. And when I compare the amount of DR that I have on endothelial cells, it is equivalent almost to the B cells that we transform and use as markers for expression of HLA class 2. It's not trivial. It's there in high amounts. And so I haven't, you may get a little bit up, but it's not, it's robust to begin with.
So the regulation is totally different than the classical endothelial cells.
Pardon?
The regulation is different than what we consider a classical endothelial cell, where the expression is only upgraded by inflammation.
Right. People have ignored this. It's not in the textbooks. Nobody pays attention to this because it's human and it's very specific and it's something we shouldn't be ignoring.
No, I don't think
but
so.
it's there. And, if you give enough interferon to any cell, it'll upregulate class 2. But again, I had spent a fair amount of time and the amount of interferon is it's trivial. It's what you measure in any of us. And it's there all the time. And it's there all the time, 1,000 kidneys later.
Well, thank you again, Kim, for a really compelling talk. There's a question from Roberto and Goyang in the chat if you're able to access. And we will
okay.
to our
thank you.
Yeah, thank you.